Where is DNA located? How is DNA packed into a cell? What is a chromosome? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Krajnovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. Where is the genetic information stored in a cell? The answer to this question depends on whether we are talking about viruses or bacteria, or about organisms that already have a cell nucleus, such as fungi, plants, or animals. We already know that hereditary information is encoded in DNA molecules. We have already talked about them earlier. In the context of discussing DNA, let's pause to consider its remarkable linearity. What do I mean by that? We can define linearity as the ratio of one dimension to the other two dimensions. For example, the complex human DNA contained in each of our cells measures a total of 2 meters. Meanwhile, the diameter of a DNA molecule is only 2 nanometers, that is 2 billionths of a meter. If we were to project this DNA into scales more tangible to us and imagine the DNA as a climbing rope 1 centimeter thick, its length would be 10,000 kilometers, which is roughly a quarter of the Earth's circumference at the equator. Let's not forget that cells are usually only tens of micrometers in size, which is millionths of a meter. It's clear that there must be some way to cram such long DNA molecules into cells. Even the simplest organisms, including bacteria and viruses, have developed strategies that allow them to pack long DNA molecules into their cells, or in the case of viruses, into their viral particles. The strategies for achieving this differ between bacteria and eukaryotic organisms, but there are general principles. Proteins always assist in the compaction of DNA. Bacteria do not have a nucleus, which is why they are referred to as prokaryotic organisms. Their genome is located freely in the cytoplasm of the bacterial cell. The genome of bacteria is represented by a single double-stranded DNA molecule known as the bacterial chromosome. Most often, it is a large circular DNA molecule that is millions of nucleotides long. In bacteria, the genetic material, DNA, is intricately coiled into many distinct loops, which is an exceptionally ideal and efficient way to compactly pack the circular DNA into a very small, confined space within the cell. In addition to this large DNA molecule, some bacteria also contain small, usually circular DNA molecules called plasmids. For example, they encode bacterial resistance to antibiotics. Many plasmids can be easily transferred from one cell to another and can even move between different bacterial species. Different bacterial species can therefore share a sort of gene bank or pool present on plasmids, which they can borrow from each other as needed. The emergence of the cell nucleus as a place to store genetic information was a major evolutionary innovation. Organisms such as fungi, animals, and plants are therefore referred to as eukaryotic, meaning they have a true nucleus. In the nucleus, DNA, together with proteins, forms complex structures called chromosomes. Each chromosome contains a single DNA molecule. The eukaryotic genome is therefore fragmented. Who knows? Maybe it's a kind of memory of the first genomes made up of many shorter DNA molecules. I've talked about that before. Although the genetic information of a eukaryotic cell is found mainly in the nucleus, we must not forget about mitochondria and, in plants, also chloroplasts. These organelles contain circular DNA molecules, evidence of their ancient bacterial origin. But that's a topic for another time. Let's get back to chromosomes. Proteins bound to DNA ensure that the spatial structure of chromosomes changes during the cell's life. For most of the cell's life, chromosomes are relaxed and more or less evenly fill the nucleus. At the moment of cell division, they begin to change. The chromosomes shorten and thicken. They form the characteristic chromosome structure as we know it from microscopic images and textbooks. The chromosome then splits lengthwise into 2 identical pieces, called chromatids, each containing one of the newly copied DNA molecules, thus ensuring the even distribution of genetic information into both newly forming daughter cells. Our genetic information is divided into 46 chromosomes. We actually have 23 pairs of chromosomes, one set from our father and one from our mother. Even though the information from both parents differs from each other a little, in this way, the genetic information of all sexually reproducing organisms is elegantly backed up. It's as if we had two bookshelves at home in the living room, where the books and their order on the shelves are identical. Only some of the letters in the texts of the books will be changed here and there. Our closest relatives, chimpanzees and gorillas, have one more pair of chromosomes than we do. And what about our pets? A dog has 78 chromosomes, a cat 38, and a rabbit 44 chromosomes. However, there is no correlation between the number of chromosomes and the complexity of an organism. Extremes in chromosome numbers include, for example, the ant Myrmecia pilosula, which has only one pair of chromosomes, with the male carrying just a single chromosome. Or, on the other hand, there is a species of fern that has 630 pairs of chromosomes. That's all for today. In the next episode, we'll talk about the size of genomes in various organisms and what the C-value paradox is. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. That truly helps these videos reach a much wider audience of people. I really do appreciate all of your continued support. Thank you and bye-bye.